I'd like to begin by acknowledging the owners and traditional custodians of the land in which we meet today, the Wurundjeri people of the Kulin Nation, and I pay my respects to their elders past, present and emerging. So today, you're going to hear an amazing talk by Sophia Davidson. She did her undergraduate training at the University of Newcastle and honours there with people who you may be familiar with who work on asthmatic lung disease, and her project was on the role of, as you'll hear, type 1 interferons, which are a passion of hers, and the role of TLR7 in that example. And then she went and performed her initially as a research assistant and then as a PhD student at what was then Mill Hill and turned into the Francis Crick Institute in London, working on, again, type 1 interferons, and in that case, the role of influenza. And of course, when she wanted to do a postdoc back here in Australia, it was going to be on type 1 interferons, and that has indeed been the case. So I said that it's a wonderful story she's going to present to you today, and don't just take my word for it. This work has been recognised. She is an NHMRC ECF Fellow, and she is also the Christina Fleischmann Award winner based on this work. And what that basically means is that it was the best work submitted to the International Society for Interferons and Cytokine Research last year, presented by a female scientist. So it really is top caliber stuff. So I hope you're excited about it, because she gives a great talk as well, and I'll leave her to show you the details.
I love a Seth Masters introduction. So today I'm going to talk to you about, autoinflammatory diseases. And in the most basic of terms, an autoinflammatory disease is a disease driven by inborn genetic mutations that result in the dysregulation of the innate immune system. Patients suffer periodic episodes of fever, and this comes along with induction of non-specific inflammation of patient organs. And this inflammation is incredibly damaging. If left unchecked, it can lead to tissue damage and disease and even disability. So there are over 50 different autoinflammatory diseases recognised to date. And because that's such a huge number, and also because we're scientists and we like to categorise things, we separate them and categorise them based on the driving inflammatory pathways. So inflammasomopathies are driven by inappropriate activation of inflammasomes, and they're normally treated by blocking the by the IL-1 family of cytokines. Irelopathies are driven by inappropriate activation of NF-kappa B, and interferonopathies are driven by aberrant type 1 interferon signaling. And the autoinflammatory disease that I'm going to talk to you about today is an interferonopathy. So proteasome-associated autoinflammatory syndrome, or PRAS, is an incredibly awful autoinflammatory disease. Patients suffer these episodes of flare where they have terrible skin rashes, and the systemic inflammation over time results in muscle wastage, redistribution of fat, loss of fat, basal ganglia calcification, and splenomegaly. And all of this is to really emphasize the point that these kids have a really high burden of disease. As the name implies, PRAS is driven by loss of function mutations, which can either be homozygous or can be compound heterozygous, but these loss of function mutations are all in genes which either code for proteasomal subunits or the proteasome maturation complex. Before we go on, just a gentle reminder of what the proteasome is. It's essentially your cell's recycling. Bin. So the ubiquitin proteasome system is one of the key degradation pathways that your cell uses to get rid of unwanted proteins. And what happens is a cell decides it doesn't want a protein anymore. It tags it with ubiquitin. And this ubiquitin tag allows for this unwanted protein to be recognized by the regulatory cap of the proteasome. The unwanted protein is then unfolded, fed into the catalytic core of the proteasome, where it's digested into peptides and spat out into the cell to be recycled. And this system is not only important for the removal of misfolded or damaged proteins, it's also important for the removal of proteins of pathogen origin, say when you have a viral infection. And the proteasome also degrades short-lived proteins in order to allow normal signaling cascades in the cell to occur. So, the proteasome is integral for cell signaling, cycling, and general homeostasis, as well as cell defense. And given this is such a complex and important piece of machinery for a cell, you then have to wonder, what happens when it's not working? Well, if we look in our PRAS patient samples, you can see that these are whole cell lysates, and this figure's been taken by Bremen Associates. And essentially what they've done is they've looked for catalytic activity of the proteasome. And this is where it should be, and our PRAS patients are highlighted in red. And as you can see, regardless of the mutation, the net result is a decrease in function. So you have a decreased functionality of your proteasome. And perhaps unsurprisingly, this results in a buildup of ubiquitinated proteins. So we can see this in transformed patient B cells, which have been run on a Western blot, and we've got 2 controls and the PRAS sample here. And you can see this huge smear of ubiquitin staining. And this smear of ubiquitin staining is representative of all the proteins which have been marked by the cell for degradation, but haven't been degraded because the proteasome is not functional or has a decreased level of functionality. They're essentially Brexit proteins. They said they were going to they said they were going to leave. They were tagged with ubiquitin, and yet still they're here, and they remain. We can also see this ex vivo. If we look in patient skin biopsies, you can see this heavy ubiquitin staining in brown. And this is due to this loss of function of the proteasome, and it's not because of an inflammatory disease, because if we look in a psoriasis control, you don't see this buildup of ubiquitinated proteins. And the thing that got me really interested in PRAS to begin with was that along with this buildup of polyubiquitinated proteins, there is increased secretion of pro-inflammatory cytokines, including NF-kappa B cytokines like IL-6 in PRAS patient blood. And on top of this, there's also a type 1 interferon gene signature in the blood. And as Seth told you in his introduction, I really like type 1 interferons. But it's kind of weird, right? Because type 1 interferons, when you think of them being induced downstream of a viral infection. They're normally induced in response to stray nucleic acids, not the buildup of protein trash. In case you don't think of type 1 interferons, here's a simple introductory slide to them. Basically, they're a family of pro-inflammatory cytokines. There's always one interferon beta, and then there's a handful of interferon alpha subtypes. And they were discovered and primarily characterized in the context of viral infections, but their expression can be induced by a range of stimuli. So what happens is this stimuli activates a pattern recognition receptor, and this induces a signaling cascade which results in the phosphorylation of IRF3 and interferon beta, and to a lesser extent, interferon alpha expression. When interferon alpha and beta are secreted, they act in either an autocrine or paracrine manner, and they bind to a single receptor. So it doesn't matter if it's an alpha or a beta, they all bind to the interferon alpha beta receptor. Ligand binding to this receptor results in a signaling cascade, which involves JAK1 phosphorylation and activation of STAT1, which is also phosphorylated. And the activation of STAT1 and this complex results in gene transcription of a whole suite of genes known as interferon-stimulated genes. So this is hundreds of different genes which are activated by this single inflammatory cytokine family, which have a myriad of jobs. A lot of them are cell intrinsic defense, a lot of them code for cell intrinsic defenses, such as MX1, but some also encode for chemokines or cytokines, including CXCL10. And CXCL10, like many chemokines, is an inflammatory chemokine in itself. So in this way, by inducing potent immunomodulators, type 1 interferons provide a feed forward of inflammation in the system. So given this huge suite of genes, which is induced downstream of type 1 interferons, and the fact that they only use one receptor, and this receptor is expressed on basically every cell type in your body, it's probably unsurprising that type 1 interferons drive such a dramatic autoinflammatory disease like PRAS. And to kind of really emphasize how strongly type 1 interferons drive disease in PRAS, patients are now being treated with JAK1 and 2 inhibitors. So by inhibiting JAK1, you can block the downstream signaling and the induction of all these pro-inflammatory factors, which are interferon-stimulated genes. So this treatment works pretty well, but as you'll remember from a couple of weeks ago from Isa's talk, JAK1 and 2 aren't just downstream of type 1 interferon signaling. They're downstream of lots of other pro-inflammatory cytokines, and so this is a highly immunoregulatory drug protocol to be put on. So there's no targeted treatment for PRAS patients. Okay, so now we're all incredibly interested in PRAS. And if you're interested in a disease, obviously you want to study it. And the first thing you need to study a disease is a model. So being in the master's lab, I used CRISPR. And using CRISPR-Cas9 gene editing, I deleted 2 genes which are known to cause PRAS and that was PSMB8, which codes for I-beta5, and PSMB9, which codes for I-beta1. And these are both proteasomal subunits. And what was really nice to see is when we deleted these proteasomal subunits, the net result was an increase in ubiquitinated proteins. And this buildup of polyubiquitinated proteins correlated with elevated type 1 interferon signaling. So if we look at the protein level, you can see that when I intracellularly stain for interferon alpha and run this on a flow cytometry machine, there is increased levels of interferon alpha in our PRAS model cells that I don't see in my parental THP1 cell line. And this is just an example histogram, and it's quantitated here. If I look for interferon beta, and the easiest way to do this is by transcription, we can see that when we look for interferon beta mRNA, this is elevated in the PRAS model cells as compared to the parental controls. So we've got induction of type 1 interferons themselves. On top of this, we know that type 1 interferon is signaling because we see phosphorylation, meaning activation of STAT1, which we know is downstream of the type 1 interferon receptor. And we also see expression at baseline of interferon-stimulated genes. And here I'm just showing you MX1 and ISG15. We can also see this at the protein level by looking for CXCL10, a chemokine induced by type 1 interferons. And you can see that we can test by Eliza that this is secreted into the supernatant in PRAS model cell lines significantly more than it is into the THP1s. We can also see that NF-kappa-driven cytokines are also being transcribed. You can see here TNF and IL-6 are both elevated as compared in the PRAS model cells as compared to the THP1 parental controls. And this all fits with what we see in the patients. All of these hallmarks, the inflammatory signaling profile, and this buildup of ubiquitinated proteins were all observed and published in PRAS patient samples. And so these two events must be intimately linked. And to kind of really drive this point home, we can even enhance this phenotype by driving the buildup of ubiquitinated proteins. So we use a non-specific DUB inhibitor known as PR619, and this unbiasedly drives the buildup of ubiquitinated proteins. And you see when we do this, we can massively elevate the inflammatory signaling gene profile that we have, particularly in the PRAS model cells. So the buildup of polyubiquitinated proteins is driving type 1 interferon. And as I mentioned before, this is kind of weird because normally interferon alpha and beta are induced by viral infections, by the activation of a pattern recognition receptor. But our cells have pattern recognition receptors for all kinds of things, not only for viruses, but also for bacterial infection, for calcium flux, everything. And so we began to wonder, given that the buildup of unwanted proteins surely must cause the cell damage at some point, it must be a danger signal. So surely there must be some way for a cell to recognize that there's a buildup of unwanted proteins. Surely there must be a pattern recognition receptor for ubiquitinated proteins. And to get at this, we decided to look at known pattern recognition receptors which have been documented to drive a potent amount of type 1 interferons and NF-kappa B signaling. And just to introduce you to the usual suspects, there are the endosomal TLRs, which rely on UNC93B, a chaperone protein, to locate them into the right place so that they can signal. There are the cytosolic RNA detectors. These include the RIG-I-like receptors, which absolutely require MAVs in order to induce signaling downstream, and PKR. And there is the CGAS, and the CGAS, which is a cytosolic DNA sensor. And it uses STING to drive an inflammatory signaling pathway. So when I put this slide together, it kind of struck me how hard the job of CGAS is. It's essentially in a bag which has a huge amount of DNA in it, but it can only respond to pathogenic DNA, the presence of pathogenic DNA. So how does it recognize the difference between pathogenic DNA and what's safe to be around? Well, CGAS is localized in the cytosol. So any DNA that's in the cytosol is considered a pathogenic threat. And I thought that's quite clever, that you use physical separation in order to determine whether or not a particular molecule, say DNA, is pathogenic. So we have all these different receptors for DNA and RNA, most of which are cytosolic. So maybe one of these could be sensing the buildup of aberrant proteins. And to get at this in a really quick and dirty manner, I used two chemical proteasome inhibitors, delanzazib and oprozomib. And you can see if I treat THP1s with these proteasome inhibitors, that they induce type 1 interferon beta transcription in a really titratable manner. Armed with these inhibitors, I did my own form of a very rough and dirty CRISPR screen, very rough CRISPR, not even a CRISPR screen, a very rough screen. And basically I took these inhibitors and I threw them on any cell type that was a knockout that I thought was relevant and I could get my hands on. And very quickly, I excluded endosomal TLRs because the deficiency of UNC93B did not alter the induction of interferon beta in response to oprozomib treatment. I also excluded RIG-I-like receptors and CGAS because cells deficient for their downstream signaling molecules had no issue in inducing interferon beta transcription in response to either delanzazib or oprozomib. And at this point, I'm kind of running out of cytosolic receptors, but happily, I got my hands on some PKR-deficient mouse embryonic fibroblasts, and as you can see, in these cells, treatment with proteasome inhibitors, regardless of which one it is, doesn't induce the transcription of interferon beta. And this wasn't just true in the true in the mouse embryonic fibroblasts. When I deleted PKR from my THP1 cells, I saw the same thing. So you have induction of interferon beta transcription when you treat with the proteasome inhibitor, and this doesn't occur when the cells are deficient for PKR. And this is not something that's due to a general decrease in the ability of the cell to drive interferon beta transcription, because if we treat the cells with CGAM, which activates STING to drive type 1 interferon signaling, we see that our PKR deficient cells respond comparably by way of interferon beta induction as to what we see in the parental THP1 cells. Okay, so this was really cool, but you know, it's chemical inhibition. So I went back to my PRAS model cell lines, and it was really gratifying to see that in these PRAS model cells, we have phosphorylation of PKR at baseline as compared to what we see in the parental line. And phosphorylation of PKR indicates activation. Furthermore, if I delete PKR, from my Pras model cell lines, I can entirely blunt aberrant interferon signaling. So you can see when we treat with the PR619 to enhance the phenotype, we go from a level of interferon beta induction, which is clearly pathogenic and way up here, to all the way down here when we take away PKR. Similarly, we see the same blunting in IL-6 induction, and also we see a blunting of ISGs, which you do expect since they are downstream of type 1 interferon signaling. Furthermore, and kind of maybe more importantly in the context of treatments, if we use a PKR-specific inhibitor, known as C16, we can again entirely blunt this inflammatory signaling. So here, there's our Pras model cells, and we treat them with the PKR inhibitor, and this aberrant interferon beta expression is significantly ameliorated. And again, the same for IL-6 and MX1. So, what is PKR? Well, protein kinase R is a pattern recognition receptor known to be activated by double-stranded RNA. Upon activation, PKR forms a dimer and induces signaling cascades which activate NF-kappa B and the transcription of type 1 interferons. And what makes it such an appealing candidate in the context of proteotoxic stress is that PKR is also a serine threonine kinase, and one of its downstream substrates is EIF2 alpha. So when PKR is activated, it phosphorylates EIF2 alpha, and phosphorylation of EIF2 alpha leads to an inhibition of protein translation. So this makes a lot of sense. If you have a buildup of polyubiquitinated proteins, your proteasome is clearly not working properly. So your cell activates PKR, this inhibits protein translation, and hopefully this gives a break to the proteasome, allows it to catch up and restore the cell to homeostasis. And in case you missed it, because I didn't point it out before, if we look in our Pras model cells, we have phosphorylation of EIF2 alpha, which matches the phosphorylation of PKR. So, we may very well have a sensor for proteotoxic stress, and this could be PKR. But what is it sensing? So PKR is known to be activated by double-stranded RNA. It has these two double-stranded RNA binding motifs. And recently, there was a paper that came out from Kim and Associates that's found that PKR could be activated by endogenous RNA. And the vast majority of this RNA was from mitochondria. So we began to hypothesize. Maybe the buildup of ubiquitinated proteins was causing stress on the cell and was leading to the creation of reactive oxygen species, and somehow this was damaging the mitochondria and causing the release of mitochondrial RNA. So it was a hypothesis, and we decided to test it. And in order to test it, we took a leaf out of the Kyle Labs book, and we used ethidium bromide culturing in order to deplete mitochondrial DNA away. So basically, we took our THP1 cells and cultured them for 3 weeks with a low level of ethidium bromide. And this results in the depletion of mitochondrial DNA. So if we look at TFAM, which you can basically think of as the mitochondrial DNA's histone, this is entirely depleted away. And when we look for mitochondrial RNA gene expression, we see that this is significantly ablated. And if we normalize this to RNA expression from nuclear genes, you can see that we've essentially entirely lost mitochondrial genomic material in these cells. So we have cells which are depleted from all mitochondrial genome, and we have cells which have a functional mitochondrial DNA. So if mitochondrial RNA is activating PKR under conditions of proteotoxic stress, these cells should exhibit no inflammatory signaling when we treat them with a proteasome inhibitor. However, this was not the case. So as you can see, cells that, whether or not the cells have mitochondrial DNA and therefore RNA, it doesn't matter. They still induce interferon beta and IL-6 to a comparable level. So, PKR is not being activated by mitochondrial RNA in PRAS. But if it is an endogenous RNA, well, what is it? Well, the fun thing about double-stranded RNA binding motifs is they don't just bind to double-stranded RNA. Instead, if you start looking around in the literature, you can find a handful of proteins which are known to interact directly with PKR. And so I collected these proteins and I performed my own little CRISPR screen where I knocked them out and then treated with a proteasome inhibitor in my THP1 cell line. And I only got one hit, and kind of surprisingly, it was not PACT, which is probably the most well-characterized protein interactor with PKR. It was not TAR RNA binding protein, which has its own double-stranded RNA binding motif. It was actually interleukin-24, which is a cytokine. But as you can see, if I treat my cells with the various knockouts of various genes with the proteasome inhibitor, it's only in the IL-24 knockouts do we see a blunting of interferon beta induction. And this is not due to a general dysfunction of these cells because we knocked out IL-24 or any effect on induction of the interferon pathway, because if we activate the CGAS signaling pathway using poly-D-ADT, you see that the IL-24 cells respond just the same as the other knockouts in the experiment, as well as the parental controls. So I took this data to Seth, and I was like, you know, Seth, I think it's IL-24. I think IL-24 is activating PKR. And he's like, as in the cytokine. I'm like, yeah. He's like, I think you need to do some more experiments. So I did. And one of the things I did was I re-expressed IL-24. So I took my knockouts, and I put IL-24 back in, and as you can see very nicely, when IL-24 is not present, we have induction of interferon beta transcription upon proteasome inhibition, and it is decreased in my IL-24 knockouts, and this can be rescued when I add the IL-24 construct back in. So, a slide on IL-24. It's a member of the IL-10 cytokine family, and it is a cytokine, so it signals through two heterodimer receptors, both of which involve the IL-20 receptor subunit B. It's thought to mostly be made by immune cells, such as monocytes and T cells, but generally the cells that respond to it are stromal cells like skin cells. And the reason why I started becoming interested in IL-24 to begin with, and the reason why I actually knocked it out, was because when it was originally discovered, it came out in a cancer screen. And the kind of overexpression studies that were done afterwards on this protein found that it could interact directly with PKR, and quite tantalizingly, it could be degraded away from the cytosol by the proteasome. So these studies were both really tantalizing and exciting to read given this data. But, you know, I'm working with chemical inhibition. Did it actually repeat in my PRAS model cells? So I performed intracellular staining on my PRAS model cells, and quite nicely, you can see that there is increased level of IL-24 protein in these cells. So this is an example histogram, and it's quantified here. On top of this, and really excitingly, if I delete IL-24 from my PRAS model cell lines, I ablate activation of PKR, and concomitantly, this results in a reduction in the activation of eIF2 alpha and STAT1. So without activation of PKR, it then follows that in my PRAS model cells where IL-24 is deleted, we see a blunting of interferon beta induction, and of course, a reduced induction of interferon-stimulated genes. So here I'm just showing you CXCL10 secretion. Cool. So now we have two pieces of the puzzle. We have IL-24 and PKR, but we don't know how they're interacting. How is IL-24 activating PKR? And given that it's a cytokine, the kind of obvious question was to ask, well, is it inducing a signaling cascade that's activating PKR? So IL-24, as I mentioned before, can signal through two receptors, both of which involve the IL-20 receptor subunit B. So we deleted this receptor subunit, and it didn't impede induction of inflammatory genes in the context of proteasome inhibition at all, really. So if it wasn't acting extracellularly, could it be acting intracellularly? So I repeated the experiment that Petir and Associates previously published in my PRAS model cells. And really nicely, we saw that in my wild-type PRAS model cells, if I immunoprecipitate out PKR, IL-24 comes down with it in complex. And full disclosure here, I'm using I'm using cells which have IL-24 expressed with a HA tag in them. And essentially I'm doing that because, as you may have noticed on previous slides, the IL-24 antibody is not good for Western blot. And for the next set of data I want to show you, I'll be using Western blots and immunofluorescence. So I needed some way to be able to identify IL-24. Cool. So the next question became, if IL-24 is interacting directly with PKR, how could this be? Because IL-24 is a cytokine, it has to follow the secretory pathway. This means it's synthesized in the ER, and here it's folded, and then here in the ER it's folded, and then it moves through to the Golgi, and then directly out of the cell or into secretory vesicles. At no point in time does it end up in the cytosol. And PKR is a cytosolic protein. So when are they getting the opportunity to meet? To look at this, I differentiated my THP1s into macrophages and then stained them and looked at them under the fluorescent microscope. And as you can see, at baseline, with no inhibitor treatment, IL-24 tends to kind of cluster around the nucleus, which is essentially where we expect the endoplasmic reticulum to be. But if I treat with delanzazib, we see that IL-24 seems to just flood out the entire cell. So we get increased presence of it, and it seems to be everywhere as soon as we inhibit the proteasome. So it looks like IL-24 is escaping the ER and getting into the cytosol. So I went back and stained with an ER marker called KDEL, and as you can see, while in our control cells, there is co-staining between IL-24 and KDEL, when we treat with the proteasome inhibitor, we see IL-24 still matching with KDEL, but then also speckling other parts of the cell, and we think this is an escape out into the cytosol. And all of a sudden, this kind of makes sense, right? Cytokines like IL-24 are meant to be secreted. So if IL-24 ends up in the cytosol, like here, then it's mislocated. It must be, and one of the principles from pattern recognition receptors is they recognize molecules that are in the wrong place. So a cytokine being found in the cytosol is a danger signal because this protein is mislocalized. And when a pattern recognition receptor such as PKR sees a danger signal, it drives inflammation. Cool. But still, we're kind of wondering, right? How does IL-24 actually end up in the cytosol? Well, not to be very patronizing to the younger members of the audience, but when you can't solve an experiment by solve a problem by experiment, have a look in the literature. So I found this paper, and it's a really nice paper, and it identified 3 glycosylation sites on IL-24. And these glycosylation sites were absolutely required for the secretion of IL-24 out of the cell into the supernatant. And if you mutated 1 or more of these glycosylation sites, you could reduce or even completely ablate the secretion of IL-24 from the cell. And then, when the authors looked for where their glycosylation mutants were appearing, they found them to be, rather than in the ER, as you'd expect, entirely escaped or entirely flooding the cytosol. So if your glycosylation doesn't occur properly, so if you think about IL-24, It's synthesized in the ER, and this involves the glycosylation. These glycosylation events to ensure that that IL-24 is folded correctly. And then when IL-24 is folded correctly, it can travel through the secretory pathway and be secreted by the cell. However, if these glycosylation events don't occur properly, then IL-24 is recognized as being misfolded and shunted out of the ER into the cytosol. And normally what happens here is any misfolded protein shunted via the ER-associated degradation pathway is immediately ubiquitinated. And in a healthy cell, this results in it being recognized by the proteasome and a consequent degradation of this protein. However, in our PRAS patients, the proteasome has decreased function. So IL-24 accumulates in the cytosol to the point where it activates a pattern recognition receptor, specifically PKR. And this then drives inflammation. So surely, if we can somehow trap IL-24 inside the endoplasmic reticulum, we can ablate inflammatory signaling. And to do this, we used a mannosidease 1 inhibitor called kifficine. I just call it KIF. So mannosidease 1 is the enzyme which shunts misfolded proteins into the ER degradation pathway. And as you can see, if we just treat normal IL-24 expressing cells with KIF, nothing really happens. But when we treat with the proteasome inhibitor, we again see this escape of IL-24 into the cytosol, which we can inhibit with treatment of this mannosidease 1 inhibitor. So here, treatment of KIF and KIF on top of delanozib results in IL-24 staying in the ER. And if we look at the consequences of this in terms of inflammation, we see that treatment with kifficine strongly blunts the induction of interferon beta and IL-6 in response to treatment with a proteasome inhibitor. So this is really new data and really exciting to get. But it's all done with inhibitors, and it's done in a fairly artificial way. So the question now becomes, how would this happen in real life, and why would it happen in real life? So to kind of get at this, I again went back to the literature, and it's interesting to find out that there are multiple alternative spliced isoforms of IL-24. And these splice isoforms appear due to splicing by the splicing factor SRP55. And SRP55 activity is induced by cell stress. And essentially what happens is when SRP55 performs splicing, you get all these shorter isoforms of IL-24. The most well-characterized of which is IL-24 delta exon 5. And the reason why we found this isoform so interesting is because in exon 5, you have a glycosylation site and also lysine 123. So lysine 123 has previously been identified to be the key lysine which is tagged with ubiquitin to degrade IL-24 from the cytosol. So we were really interested in this in delta exon 5. Given that it lost a single glycosylation site, it may have the potential to be more likely to be misfolded. And because it didn't have this lysine, it may be more stable it could be more stable in the cytosol after it's been kicked out of the ER. So I made constructs for both full-length IL-24 and delta exon 5. And as you can see, when I overexpress them in hex and run them on a Western blot, you can see this difference in glycosylation because of the difference in banding patterns. And more excitingly, when I stably express them in my THP1 cells, you can see a baseline delta exon 5 accumulates more aggressively in the cytosol of these THP1 cells. Furthermore, this can be enhanced when we add the proteasome inhibitor on. So again, full length, you don't get much accumulation at baseline, but when you add the proteasome inhibitor, this is enhanced, and you see this is massively so in the delta exon 5 IL-24 expressing cells. So this is really cool because it firstly shows a mechanism where a naturally occurring isoform of IL-24 can aggressively accumulate in a cytosol. And it also tells us that we don't really understand how IL-24 is being degraded from the cytosol, because clearly it's more than just the single lysine residue which can be tagged with ubiquitin to drive degradation. Cool. So you've got all that. In case you've been sleeping, I will summarize because the next part is all the really exciting data. So basically what we have is a situation in PRAS where you've got proteasome dysfunction, and that leads to the accumulation of IL-24 in the cytosol. Because IL-24 is a cytokine, it's mislocalized here and considered to be a danger signal. It activates a pattern recognition receptor known as PKR, and PKR acts as a cellular rheostat in that its activation phosphorylates eaf2 alpha, and this inhibits protein translation. It also drives inflammation, and this induction of inflammation then drives proteasome-associated autoinflammatory syndrome. So I'm a big fan of this pathway, but it was all worked out in immortalized cell lines using CRISPR and a lot of chemical inhibitors. How does that actually relate to real life? Well, to get at this, Seth sent me to the lab of Rafaela Goldbach-Mansky, and she's a clinician scientist that specializes in autoinflammatory diseases. And she was incredibly generous. She let me work in her lab for a few months, and she let me work on really precious patient samples. And it was really rewarding because what we found was, if I looked in patient fibroblasts, again, I could see phosphorylation, so activation of PKR, and phosphorylation of its downstream substrate, eif2 alpha, at baseline in our PRAS fibroblasts, but not in healthy controls. And I could also see increased levels of IL-24 in these PRAS fibroblasts as compared to the healthy control. If I used PRAS PBMC samples, I could see activation of PKR and also activation of STAT1 and eif2 alpha at baseline. And what was really rewarding was when I treated these cells with C16, so the PKR inhibitor, this blocked the activation of PKR, and concomitantly, there was a decrease in eif2 alpha and STAT1 phosphorylation. And of course, we saw none of these biomarkers in the healthy control. So this really demonstrates that in PRAS primary patient cells, phospho-PKR is upstream of inflammatory signaling. What's also nice to see is that IL-24, which is upstream of PKR in our pathway, is not changed by the inhibitor, really demonstrating that it is upstream of PKR. Furthermore, only 8 hours of treatment with the PKR inhibitor was enough to decrease type 1 interferon inflammatory signaling in the PRAS patient cells. So here are PRAS PBMCs, and I've just done qPCRs for interferon beta, interferon alpha 1, and this down here is an interferon score, which is essentially an aggregate of the expression of 32 different ISGs. And so as you can see, when we treat with the PKR inhibitor, we entirely blunt type 1 interferon signaling and therefore interferon-stimulated gene induction. And again, we don't really see an effect of C16 on the healthy controls. And to really drive the point home that this pathway is specific to PRAS, I also treated PBMCs from another interferonopathy. So SAVI is driven by autoactivating mutations in STING. And these autoactivating mutations drive a strong interferon response, and this is not inhibited in any way when I add my PKR inhibitor. So this really demonstrates that activation of PKR and consequent type 1 interferon signaling is occurring in PRAS, and this is a PRAS-specific pathway. So this was all really, really exciting, but there are a lot of caveats to working on patient samples, particularly when all of your patients are young children and they're all on immunosuppressive therapeutic regimes. So in order to kind of be able to expand our work and to get around this problem, the Masters Lab have started working on induced pluripotent stem cells. And I was really lucky in Rafaela's lab because firstly she had induced pluripotent stem cells from a PRAS patient, and just downstairs there was the lab of the Malik lab, and with the kind and very patient help of Colin Sweeney, I was trained into how to culture iPSCs, drive their differentiation into CD43 positive hemopoietic stem cells, and then their differentiation into various myeloid lineages. So the data that I have to show you is only on macrophages at the moment, and it is still quite preliminary, but I think it's really powerful because these are iPSCs generated from a healthy donor and the PRAS patient. And from the moment they were thawed, I treated them exactly the same. So they were iPSCs together, they differentiated into hemopoietic progenitors and then onto macrophages, always being treated in the same way and always being kept in a sterile environment. And from these cells, it's really nice to see you still have an interferon beta gene, interferon alpha and beta gene induction in the PRAS model, in the PRAS patient cells. And this elevated interferon beta and alpha gene transcription can again be ablated when I treat with my PKR inhibitor. Yeah, so to sum up, alongside the detection of RNA and DNA in our cytosol, we've shown that there's also an arm of the innate immune system that is directed against the accumulation of aberrant proteins. And although this pathway was delineated in an incredibly rare disease, I want you to think about your own research. This pathway involves the buildup of ubiquitinated proteins, as in so many neurodegenerative disorders such as Parkinson's or Alzheimer's. It involves proteasome dysfunction. And if you work on it, I hope you know that a lot of bacteria secrete proteasome inhibitors, and some viruses will actively try and overload the proteasome. And this pathway is also important and possibly of therapeutic benefit in cancer, as people with multiple myeloma are currently being treated with proteasome inhibitors. And I think really that's my favorite thing about working on rare autoinflammatory diseases, because the lessons that we learn from studying these diseases allow us to see pathways we never would have thought to look for. Like, who would look for an antiviral cytokine family being induced downstream of protein trash? Not me. So that's it. All that's left is the acknowledgments. Obviously, my biggest thanks go to Seth. He is a really supportive and encouraging boss to have. Like, as he introduced me, I've done really well in his lab. You know, and under his mentorship, I've not only pulled out this pathway, but, you know, I've been to international labs, I've spoken at international conferences, and I feel really privileged to be in the Masters Lab. Also because he hires such excellent people, including Alan, our senior postdoc and my iPSC co-parent, two very inspiring students, Cass and Annie, and a horde of research technicians, all of whom, both the current ones, Katja, Jacob, and Pawat, and the past ones, Kate and Dale down here, and Paul as well, although he was a PhD student before, all of them have had some hand in presenting the data that's here today. I'm so happy that I'm in the inflammation division, and I'm very grateful for the time I spent in the Goldbach-Mansky's lab, and particularly from the help I got from Colin. There's three more people I want to thank, one of which is Joe Groom, so when you pick a mentor, I think the best advice I can give you is you pick somebody who has qualities you see that you want to kind of cultivate in yourself. So Joe runs a very successful lab and has a healthy work-life balance and a family, which are all things that I want for my future. But also, she has the kindness of spirit to take time out from all of that to sit with me for very long coffee breaks, read through applications, and kind of just genuinely push me along. And I think I really appreciate it, and I think it shows a real generosity of spirit. And the last two people I want to thank are Hannah and Lynn, who, in my opinion, probably have the most difficult and frustrating jobs at WeHi. But in spite of this, when my health gave out 2 days before the Ideas grants were due, Hannah and Lynn, and Seth as well, just stepped in, and they took everything off my hands, and my Ideas grant was submitted very successfully, ultimately to be rejected. But I think the fact that I have this, that this support network is available to me, honestly, I think that's worth more than, the $400,000 I asked for. So that's it. Thank you for listening.
Fantastic. Thank you, Sophie. A few questions?
Does the areas which are most affected in the PRAS patients correlate with where IL-24 is generally expressed? So
IL-24 is pretty understudied, but if you look on the Broad Institute for IL-24 expression, you see that it's highly expressed in immune cells and fat. And what's interesting is with the PRAS patients, they have this loss of fat and lipodystrophy, so the relocation of fat. So there are overlaps, but it's not a kind of slam dunk definitive.
Is there still room for another protein which might be?
Definitely. I mean, I don't know if all cells express IL-24, but surely every cell has to be able to sense a buildup of polyubiquitinated proteins. So whether or not it's always activating PKR, I think remains an open question, and yeah.
Over here, Sophie. Interesting stuff. I mean, I wonder whether there's an alternative hypothesis as to how it works, whether instead of getting alternative splicing being the cause, I wonder whether you get a change in the start codon of IL-24, which removes the signal peptide. And this is a normal thing that occurs. And then that allows for a normal pool of IL-24 to occur, and that's normally just turned over by the proteasome. And so then what happens is as soon as the proteasome is inhibited, that natural turnover is blocked, and that is the thing that activates. And I think that seems to me more of a sort of a parsimonious idea, and that actually the deletion of the way that you have shown it, of course, is somewhat artificial because you've induced the removal of that exon.
Whether or not there's a start codon after the signal peptide?
So is there a methionine, another methionine there, or is there an alternative splice that removes the signal peptide?
So
and therefore that would induce a pool to be cytosolic all the time.
So I really like the hypothesis. I know that if you just cut off the signal peptide, then this causes more accumulation of IL-24 in the cytosol. But I've never looked to see if there's an alternative start site there, which, sounds like a great idea. I will.
Basically following on from that question, I mean, I agree with Chris. I think it's, I'm not sure of the mechanism that you've suggested, but I was wondering if you've been able to pull it down. So can you check to see if there is glycosylation, if the signal peptide has been cleaved in the appropriate place? All of these things to actually show that it has gone through the ER, maybe some glycosylation that's happened to prove that point that it has to go through the ER first.
I mean, so at the moment I'm doing cell fractionation assays to try and, work out, is it in the cytosol and if it's in the ER and so on and so forth. And definitely looking to see if it's glycosylated properly is an easy adjunct to that.
So it's hard to work out on a log scale, but it didn't look like deleting IL-24 or PKR activation completely suppressed the interferon response. So can you tell us what percentage of the response you think might be due to that pathway, and what other pathways you think might be involved, if at all?
So, at baseline, I've got to find the data. No. Moving on. So at baseline, it looks pretty clean. Nobody cares. Oh, no, sorry. At baseline, if you... There. So at baseline, it looks pretty good. From the press, just untreated cells, you see, it seems like deletion of IL-24 is quite good at ablating interferon beta induction. But again, if we kind of drive the system by the buildup of ubiquitinated proteins by using this non-specific DUB inhibitor, there's definitely something else which is inducing interferon and signaling, and it's happening in the wild-type cells as well. So, as a percentage, I don't know. I mean, we've gone from probably about 30 to less than 10. So 66%. And in terms of other pathways, the idea that it's only activating PKR to drive inflammatory signaling is kind of surprising. So I wouldn't be surprised if there is actual cell stress occurring and we are getting mitochondrial DNA release and some kind of activation of other pattern recognition receptors. Or it could be that there are other proteins which are activating PKR and I haven't knocked them out.
So we've reached 2 o'clock, but if there's any final questions, David, yes, you want quickly? Okay. I'm going to put this one in there.
All right, so a lot of patients with myeloma will get treated with proteasome inhibitors, and a number of them are innately resistant, and just about all of them will become resistant. I'm just wondering if you've observed acquired mutations in PKR or IL-24 in resistant patients for myeloma.
Nothing's been reported. We looked, but what's really nice about multiple myeloma is the old treatment for multiple myeloma was type 1 interferons. So the fact that you have an effective treatment which attacks the proteasome itself, but it also drives immunomodulation through the induction of interferons, I think is really interesting.
And on that note, let's thank Sophia for a wonderful talk.